Dear colleagues, let me introduce my presentation, a molecular identification of trematodes of the genus Dipostome in limnid snails from Arctic lakes. The object of the study was molecules the Limnidae family, better known as the pond snails. There are widespread in almost all freshwater bodies from the Arctic to the Antarctic and are intermediate host for most species trematode worms, including trematodes of the genus Dipostoma. Trematodes of the genus Dipostome have a complex life cycle which includes the change of two intermediate hosts. At the first stage of development, the egg of the trematode turns into miracidium, which infects a freshwater gastropod from the Limnidae family and passes into the sporocyst or parthenoid stage. Thus, the metamorphosis of Meracidium into Sacariacus in the mollusk, which after maturation leaves the first intermediate host and subsequently infects and secondly intermediate host, the fish. Infected fish become easy prey for waterfowl, which are the final host for trematodes. We used the sequences of the second internal transcript spacer of ribosomal DNA to detect and identify trematode in mollusks. This genetic marker is located between the two ribosomal RNA 5.8S and 28S and is often used in such studies. The results show that the lengths of amplified fragment in trematodes and pond snails differ. 260, 370 in trematodes and 375, 510 in mollusks. As you can see in the figure, two regions of the ATS2 spacer were amplified from DNA samples of infected mollusks, one of which belonged to the pond snails and the other with their molecular weight infecting the mollusk trematode. To determine the trematode species, the following steps were taken. The sequence of the ATS2 spacer belonging to the parasite was extracted from the gel. This region was excised from the gel and the PCR product was isolated. The fragment isolated from the gel was prepared and sent for sequencing. As a result of sequencing, information about the nucleotide sequence of the trematoid DNA was obtained. The obtained sequences were analyzed in the BLAST program and in the Insibia Gene Bank database. As a result of study of pond snails from different geographic regions, we identified individuals infected with parthenoids of trematodes from the genus Dipostome. It can be seen from the table that the same species of trematodes is found in areas that are wide remote from each other, such as Dipostomes passatium in the Archangelsk region, Buryatia and China, or Dipostome bayer in Chukotka and the Murmansk region. Moreover, a specific binding to the host mollusks of the same species, i.e. species specificity, was not found. The same species of trematodes is found in pond snails of different species and even genera. Also, during the study, a phylogeographic analysis was carried out by constructing a median network of haplotypes, the results of which are shown in the figure. The haplotype set is built on the basis of an array of 93 ATS2 nucleotide sequences obtained by us and taken from GenBank. The size of the circles is proportional to the number of identical nucleotide sequences. The numbers indicate the number of nucleotide substitutions between haplotypes. Haplotypes obtained in the course of this study are highlighted in black. This is the course of the study. Using molecular genetic methods, the presence of two species of trematodes from the genus Diplostome in the Arctic worms was confirmed Diplostome baerii and Diplostome spessatium. It has been shown that the Diplostome trematodes chose representatives of the Limnaeidae family as the first intermediate host without exhibiting species specificity to the host mollusk. The results obtained indicate that the range of Diplostome spessatium covers Siberia, Central Asia, and Europe. In connection with the above, the study of the taxonomic composition of trematodes and monitoring of their diversity in the Arctic territories is becoming an important aspect in the control and prevention of infection in humans and animals. Additionally, it is necessary to assess the species composition of the intermediate and final host. Thank you for your attention.